I'm Mark Belke. I'm the Chief Scientific Officer for Integrated DNA Technologies. We've been part of Danaher now for 4.5 years. Today, I have the interesting task of introducing somebody who really doesn't need to be introduced. I'm going to introduce Professor George Church. And George is the Robert Winthrop Professor of Genetics at Harvard Medical School. He's also a member of the faculty for the Health Science and Technology Joint Program between Harvard and MIT. And he's a founding member of the Wyss Institute for Biologically Inspired Engineering at Harvard. He serves as a member of the Bulletin of Atomic Scientists Board of Sponsors, which was originally founded by Albert Einstein. And in 2017, Time magazine listed him among the 100 most influential people in the world. And last year, he was featured among the most influential people in biopharma by Fierce Pharma. So anyway, without further ado, I would like to introduce Professor Church, who will give the next lecture, and following which we'll have a short Q&A session before we move on to the next panel discussion. So, Professor Church.
Thank you very much. It's wonderful to be talking with you, and looking forward to the panel later on and the Q&A in between. Maybe I could have the slides up here. I was given the topic of convergent innovation in genomic medicines. I often go with the given title and see what I can make of it rather than choosing my own. And that's what we're doing here today. Excited about this point of view. I'll say thank yous on almost every single slide. And there's a thank you here. And there's also a full conflict of interest website, if you're interested in that, in the lower right there. And this 50 years refers to the transfer RNA. I've been working on almost exactly 5 decades, and I'll mention a few payoffs that are happening as a way of indicating this idea of convergent innovation. So in those years, we've got the tRNA itself as a therapeutic in a company that's my alumni started, called Shape Therapeutics. The modified bases have inspired messenger RNA modifications, messenger RNA that uses the tRNA modification. Like N+1, and then expanded protein alphabet for serum stability and so forth in Grow Bio. Some of these are making their way into or through clinical trials. So the convergent innovation we'll say are combinations of technologies, sometimes from very different fields, and I'll illustrate those in green here, and social processes. Sometimes you need to do social hacking engineering in addition to the science and technology. And so this produces new products and services and so forth. So I'll just give 5 quick examples of the technology combinations, and then the next slide will be on social. So for genome recoding, we're taking this tRNA, my long love with tRNA, and putting it together with things like sequencing and virology. Nanopore sequencing was, at the time, in the 80s, when I first started working on this, was a crazy idea of looking at sequencing single molecules and doing it with one of the few single molecule methods at the time, which was patch clamp for neurons, and then combining that with helicase polymerases and synthetic fusions with pores. Next-gen sequencing, or fluorescent next-gen sequencing, was a combination of microscopy and molecular barcodes. Enhanced organs was editing, virology, immunology, all put together. And then for endangered systems, we're putting together ancient DNA with ecosystem planning and and animal cloning, not recombinant cloning, but animal cloning. So um, so now let's talk about social processes now in red. So that I'm going to say in the very next slide talk about a way of getting enhanced immunity and viral and resistance to all viruses. And this requires some nuanced discussion about norms, what we think is normal in terms of enhancement. A huge fraction of medicine and technology in general is enhancement if you reflect on it, but still it requires some adjustment or expectation handling. So when we have brought down the cost of sequencing 20 million fold or more, but still applying it in a wholesale manner for infectious and inherited diseases has not kept a pace with the incredible reduction in cost. And so that's partly a risk assessment issue that I'll mention. Um, gene therapies are usually talked about with rare diseases, but if we talk about it in the case of common diseases, I will argue that we can get down to incredibly low prices. And I know that the discussion a few times today has turned to how we bring down the prices, but this is potential radical change from millions of dollars per dose to single $2 a dose. And then talk about how we can use machine learning and large libraries, mega libraries, MLML. And there, the societal limitation is kind of a holdover to the times where a huge fraction of the NIH budget was hypothesis driven. This is pre-genome project, pre-NIBIB Engineering Institute. And then finally, when we start engineering ecosystems for public health, there's some counterintuitive things, both about climate change and about engineering ecosystems. So let's talk about this concept of zero viruses. There were 4 reasons that we started changing the tRNA, mostly tRNA-based genetic code. And these were genetic and metabolic isolation, non-standard amino acids, which was the basis of Grow Biosciences, as I mentioned, and this idea of zero viruses. And we started doing this, in the early 2000s, and there's a series of papers, and here's some of the people working on it: Nili Ostrov and Mark LaJoy. Dan Mandel started Grow Biosciences. So here, cut to the chase, after a few iterations where we got a large number of viruses just by changing the genetic code by 1 codon out of 64, and then by 3 codons out of 64. That was not sufficient to get all of them. In particular, a category of viruses that brings along their own transfer RNA would manage to kill even our best shot on this, and this has also benefited from work in other labs like Farron Isaacs and Jason Chen's. So the thing that finally did it, we think this will handle almost all viruses. It's hard to prove that without testing all the viruses in the world, but on paper and in our tests done so far, swapping serine for leucine at 2 of the 6 serine codons results, and these are on very strong viral promoters. Just your run-of-the-mill tRNA promoters was not quite adequate, and also avoiding viral attacking of certain tRNAs with ribonucleases. Anyway, swapping serine to leucine results in a mixture of serine and leucine at all the positions that the virus expects to have those 2 serine codons. And we tested this not just with all the viruses that we know and they're in molecular biology literature, but we went to farms and sewage and got some fresh ones, thousands of new viruses in here, characterized some of them, used them in pools or singly, and it really is resistant to these. So, and we also made it simultaneously biocontained, which I think is a good idea, because something this powerful is one of the few laboratory organisms that could actually survive, possibly thrive in the wild. And so you want to be especially careful with working such strains, even when you think you're working in the lab, because it could get out of the lab, even with good physical practice. And this is Akko, Svena, and Regan have worked on this and published it in preprint form so far. So this is hot off the presses, as will many of the things that I say be, because I'm trying to be provocative about the future and convergence. So this was done, most of this was done with alternatives to CRISPR. I know that a lot of people think that I should be focused on CRISPR, but we've things like SSEPs, single-strand annealing proteins, and these serine tyrosine integrases we've been working on before CRISPR, and we're working on them now, and they were vital to getting both the one codon out of 64 changed to synonymous codons, as well as the 3 codons, and we're now working on 7 codons. And this is thanks to Gabe and Tim and Kale, Tina, and Ben. So another component of the viruses is endogenous viruses. So not just environmental ones, but endogenous ones. And we were kind of forced to look at this because as we published our first paper on CRISPR, we got invited by three of the pioneers in the field of xenotransplantation to take a look at something that they had hoped was one gene, but now they felt might be many. And when we collected everybody's wish list that had been accumulating since 1963 with one of the first successful chimp to human xenotransplants, we found 42 edits that needed to be made, and we've made all 42 of them. In fact, we've made many different strains of pigs, many different collections of edits, and these have made their way into preclinical non-human primate trials with up to 600-day survival of organs. And so this will be quickly making it into major human clinical trials in at least 3 hospitals, Jim Markman at MGH. But these endogenous retroviruses is one of the things that seemed like would be the most daunting, but turned out it was be one of the more easy things that we did. And we had to eliminate sources of nicks and double-strand breaks if we could, as much as possible, or protect the cells with P53 inhibitors and basic fibroblast growth factor. Anyway, this Luhan Yang was was the author on all these papers and co-founder of these 2 companies. So that positive experience with the endogenous retrovirus, which were repeating occasionally mobile elements, we thought that we would aim for larger families like the long interspersed repeats, like the line elements. And then since then, we've started working on centromere signs, basically all the major categories of repetitive elements, which are not just interesting because they're repetitive, but because here's a tiny subset of all the papers that indicate some relationship with senescence and cancer. It's been hard to study these because it's been hard to sequence them and hard to edit them, but I think we've gotten a pretty good indication that we can efficiently edit it, almost all of them, and to show functionality of a multi-gene family or a repetitive element, you really need ways to get nearly all of the active ones at least. So our record right now is 24,000 edits in a single cell. We did this with the adenine to guanine deaminase, thanks to our friend David Liu, one of the co-founders with me at Editas. And he's provided us with a whole series of enzymes prior to publication, some of which we've modified a little bit. This is where we really need no nicks at all. When you're doing 24,000 edits even a nick is extremely toxic. This is Raphael, Chunting, Corey, Oscar, Khalid, and Verena. So now that brings us to libraries. So in a way, that generates libraries of cells that if you don't take it all the way to completion, you get random sets of mutations in the repetitive elements. Another category of libraries, and we pioneered synthetic DNA libraries around 2002, and this has become quite popular now after about a 10-year lag. But anyway, the human transcription factor library, expression library, is some of it is synthetic, some of it is from cDNAs. But we tried to get everything that could possibly be a DNA binding protein or nucleic acid binding protein, including some splice factors, and then look through them systematically. And now we're using machine learning tools as well to find 300 tissues type recipes, right? Recipes for getting different tissue types, cell types. And then this led to contributions to these 5 companies: GC Therapeutics, Thymune, which is about tolerance, Colossal, which is about endangered species, Rejuvenate for aging, and Gameto for, as the name implies, gametes. These have made it into these preclinical trials where, for example, oligodendrocytes, sometimes combined with excitatory neurons and endothelium, can result in remyelination that is resistant to the autoimmune attacks. So in a way, they're kind of an enhanced cell or organoid therapy. And then here, functional assays that you have restoration of function. And then Alex and Parastu published this Nature Biotech paper and formed GC Therapeutics. So back to the Convergent innovation roadmap here. Now we're talking about using sequencing for infectious and inherited diseases. And this has something to do with risk evaluation. We've improved both reading and writing DNA by 20 million fold. We're going to come soon to, can we also do this for therapies? 20 million fold would be very welcome. But it seems to be going in the opposite direction. And we would claim that this is mostly multiplexing, not parallelism or automation. It is actually having in one droplet hundreds to billions of molecules that are barcode tagged or in some other way kept, having shared chemicals and reagents. Some people forget that next-gen fluorescent next-gen sequencing is microscopy. Sometimes it's a very thin layer, so you're not getting the z-axis, but it is. And so when you try to get structural information without microscopy by going through next-gen sequencing, you're actually just using it in a different way. And by the way, Danaher played a role in this. It was the guts of 3 of the next-gen sequencing, of the original next-gen sequencing fluorescent instruments that had looked different on the outside, but had the same Danaher contents that we developed at the Wyss Institute. And then we worked with Lynx and Complete Genomics, which went on to become Illumina MGI. And then there's smaller, more portable versions of these that we've also, our most recent publication is with Roswell on molecular transistors. But we also think they're the only group that worked with both the Roche and the Oxford Nanopore form. The Roche is still not out commercially, but it is, as far as I know, it's still quite healthy. And both the molecular transistors and the Roche are distinctive in that they drop the baseline in between nucleotides. And then that leads us to spatial versions. This is taking the fluorescent sequencing back to its roots in microscopy, but now developing that z-axis so you can get thick specimens, which in principle could be more efficient because you're not taking it apart and then randomly distributing it. You're leaving everything where it was, and you can get specimens up to 40 microns thick. And at least in some cases, not so much in situ, but you can get down to 0.2 nanometer precision, more typically 20 nanometer. And the Wu lab has worked with Brooker on this, and our lab has worked with ReadCore and 10x Genomics to bring out the Xenium. Rich Terry in particular. I'm just going to quickly show you DNA, RNA, and protein. These are the maternal and paternal pieces of chromosome 19. This is just a little kind of zooming in on it. And you can see you can display it just like any other macromolecule. But these are very special macromolecules because they're, an extended form would be 2 meters long. And the Wu Lab is with whom I collaborated on the second paper, extending this to whole genome analysis at super resolution, 20 nanometers. And then it's just a blow up of a single loop where you see 10 kb region is divided up into dozens of little point fluorescence. So you can get really on the order of a few nucleosome resolution. And this is work from Near et al. And Nguyen et al. From Wu Lab. So this is RNA. Each dot is a single RNA molecule changing color, GHEC. That was a single cell, fibroblast, actually from my body. And you can the computer can then recite the series of next-gen cycles to give barcodes in situ, where you retain the three-dimensional subcellular and multicellular context. And Lee and Doherty did this initially. And then finally, DNA, RNA, protein. Protein, we put these little tags, little 9-mer tags, and do DNA exchange imaging, which is another form of super resolution imaging, where the fluorophore binds transiently. The fluorophore oligo binds transiently to the covalently oligo on the antibodies. Or we can use nanobodies, which are tiny versions of that. I'll just quickly show you an example of that, where we have conventional diffraction limited imaging of the same square as the super resolution left and right, respectively, of synapses, multiples, about 5 synapses in this picture, where the white arrows indicate the direction from pre to postsynaptic based on multiple antibodies, in this case 4 antibodies, and we can also tell whether they're excitatory or inhibitory. Probably the number of, we haven't proven this, but the number of antibodies we can simultaneously detect might have a similar limit to the number of RNA and DNA, which is probably in the tens of thousands, but that remains to be seen. Now, this is going to be a little bit odd for someone that is so involved in gene therapy, but you can see I'm also involved in sequencing, that I'm going to maintain that for rare diseases, it's more cost-effective and many times more humane to do the diagnostic route, the preventative route, as early as possible. If you have, repeated issues with embryo loss, often due to aneuploidy, then that might be IVF, be the appropriate solution to avoid the spontaneous abortions. If the earlier and earlier you go to preconception and premarital, the less impact it has in terms of false positive consequences are essentially nil. Now, that's for rare diseases. There will, then this handles de novo mutations and so forth. At least for the time being, there will be others that leak through, and we need all kinds of therapies. But orphan drugs typically, as of 1983, have been profitable. But for common disease, I think that's where the gene therapies will really shine. And there's relatively few examples so far, but I'll show you one that has greatly influenced, I think, the way we can think about this. And this is for infectious and age-related diseases using gene therapies. We have, in the context of the transcription factor library we have used for 300 recipes. I said 2 of those recipes are producing primordial germ cell-like cells and granulosa-like cells, and they're both showing progress in human, and the granulosa cells are moving towards clinical test trials for helping to mature natural oocytes in the IVF clinics. And this is Christian and Merrick. So the risk assessment that sometimes happens here is that people, individuals, even though public health officials might wrap their head around this very easily, individuals have trouble betting on risks that are sort of in the low percentage range. So, for example, climate change was something that was relatively easy to deny, even when the World Health Organization said it could affect 250,000 additional deaths per year. Seatbelts, even after it was illegal to not have them in your car and not drive with them on, it still required a special device that made an annoying sound unless you buckled up, and that was about 0.2% of the deaths per year. There have been estimates like this one from Larry Lind in the Ministry of Health of Canada that there might be millions of dollars per patient, whether or not they're taking orphan drugs for the most severe Mendelian diseases, and there might be as many as 7,000 of such diseases are individually rare, but collectively common enough that it can affect 3 to 8% of the population. Again, these low percentages, people say, well, I'm in the 99%, I don't need to worry about this. And I think that's one of the problems with uptake, even though we've got very low costs now. We've heard a little bit about cancer from Sid and others, that it's often framed as, how do we deal with metastasis? How do we deal with early detection, which really means by the time it's loose in the blood. But the ultimate is prevention. And I think a huge fraction of things that gets us many decades of high quality life are preventative. And these include the preconception, pre-implantation genetics I've already mentioned in other contexts, but also adult genetics where pre-symptomatic prophylactic surgeries can be done for breast cancer, stomach cancer, and so on. Environmental sources, smoke, UV, etc. And another environmental source that people don't always, regular folks, not you all, don't always think about is that in the infectious diseases. I've listed 11 of them here. I'm sure there are more. Probably the one that most well known by the lay audiences would be HPV, for which there's a vaccine that's effective. Which brings us to the renaissance of interest in vaccines. We've got the most famous ones are respiratory. Lyme disease is coming back. It was ruled out, I think, because of an unfortunate timing with a fake data about vaccines. But let's look in particular at the SARS-CoV-2 vaccine. In just on this topic of gene therapies, and what do the gene therapies have? The top 5 vaccines for COVID-19, I would argue, were formulated as gene therapies, independent of what they're called or the details of the immune component of it. And those 3 of them were adenoviral capsids surrounding a double-stranded DNA. And one of those is the cheapest or least expensive of the whole set, which is a little over $2 a dose. Contrast that with one of the earliest FDA-approved vaccines for a rare disease, for spinal muscular atrophy, which was $2.5 million a dose. So bring it down by a factor of 1 million, probably mostly due to the scale of that we have now. Billions of people at risk for COVID-19. And a tiny fraction of the population for these rare diseases. And this is a way of making this not just a therapy for the wealthy countries, but possibly making it available to everybody the way the smallpox is now because of extinction. So, millionfold improvement. And it got very quick approval under emergency use, only 11 months for some of these gene therapy formulations of vaccines, even though there were a whole new category in vaccines. This was not your run-of-the-mill seasonal flu update. This was a whole new category and still got approved. And I think it's established a safety and efficacy record for that should hopefully spill over into some of our considerations about gene therapies more generally. Now, I would argue that the other major category of disease that is in that same league of affecting billions of people worldwide is diseases of aging. And I'm not talking about longevity here necessarily. We're talking about diseases of aging. And there's a half empty, half full of it's fairly well understood the pathways. And I've got 10 pathways here in this augmented diagram that some of you may know about. João Pedro de Magalhães was a postdoc in my lab who, to this day, maintains this a couple of very useful aging gene databases and animal databases. And this was used by Noah Davidson and in these three papers here using combination therapy. So combination therapy you see in many other fields. Here, the three of them were the Yamanaka factors, which we co-authored with David Sinclair, but three of them were more blood-borne factors. And we know from young blood experiments and that there is something there. And rather than dissect out all the cellular, small molecule, and protein components, possibly RNA components, we just took a guess from the literature what 45 different gene therapies that might be borne by the blood, tried them individually in mice, and took three that looked promising, tried them in every possible combination in mice. And here we're not just looking at biomarkers that might be correlated, might or might not have a causal, easily understood causal component, but we also looked at multiple diseases. In our first paper, we did these top four, high-fat diet, obesity, diabetes, and cardiac damage and kidney disease, and then quickly added other diseases in the subsequent papers. And this has gone from successful mouse studies to dog clinical trials for veterinary use, and then that's looking positive enough that it will be in human clinical trials soon. And again, Noah Davidson was postdoc and then started Rejuvenate Bio, San Diego. And then we're very keen on machine learning for a variety of uses. We published 8 papers recently, since 2019, on language-based models. So this is to be distinct from 3D models, which as a crystallographer I like, but as for practical reasons, the language models seem like UniRep have been very valuable. And these 8 papers resulted in 5 companies. Nabla on antibodies, these are all postdoctoral initiated. Dino Therapeutics on capsids, Hatch on RNA regulation, Shape Therapeutics, I already mentioned, is delivering RNA therapeutics. And then Manifold is quite interesting. It's using machine learning and mega libraries to find pairs, protein peptide pairs, that have almost no off diagonals, no crosstalk, beautiful long diagonals with hundreds of these pairs, so that the one can monitor multiple protein drugs simultaneously in, say, one expensive non-human primate clinical trial, and you can test maybe thousands of drugs in a realistic scenario early on. Picking one of those 5 companies, Dino, where it was about viral capsid design, trying to find new tissue specificities better than wild type, all the different existing serotypes, and also avoiding immune problems that come from reuse of a vector or just the existence of the vector in the otherwise naive population. Pierce and Eric published these two papers and, showing that we could get tissue specificity here. Red means better than wild type at accumulating in these 6 tissues, and we did many other tissues as well, where we made every possible codon substitution at every position along the genome, the capsid. And that's doing every possible single codon change. But what if we want to do 28 amino acids in a row? How diverse can we generate that? How far can we go in sequence space? Well, with all of our previous methodology, naive models, it was hard to get more than 4 out of 28 mutations simultaneously in one round, because at that level of mutagenesis, you would get it would be fatal to the protein. Instead, when we use logistic regression or other neural net models for machine learning, we can make big libraries that have 25 out of 28 changes at sort of 90% success from the examples suggested by the machine learning. So this would be 0 with our previous models. And not only can get 25 out of 28, 26 out of 28, 27, 20, even 28 out of 28 changes, and still make a library with a noticeable number of hits in it that were predicted by the machine learning. So this, I think, is going to be generally a whole new way of doing protein design and engineering. And then finally, just a slide on ecosystems, because everybody wants to talk about this. I already mentioned the climate change cost has been estimated at the World Health Organization. And we're also interested in endangered species and endangered ecosystems, and in some case, restoration of rewilding of species, endangered species and ecosystems. And the classic example is wolves saving Yellowstone. I urge you to take a look at this if you don't already know it. The wolves didn't reduce the elk population, just kept it on the run, and that allowed the willows to grow up from little sprouts to full trees, which encouraged the beavers. Beavers changed the whole landscape, rivers and lakes, and which changed the amphibia and the fish. So you can see how one keystone species can have this effect. And we think this is true in most places where there are elephants. Elephants used to be present on all the continents except for 2. And they could conceivably help. Long story, I'm not going to go into it. Some of you may know it, about how the Arctic is such a great place for carbon sequestration, but it's also a great place for losing that carbon in the form of methane, which is 80 times worse than carbon dioxide. And here's my friend and colleague Sergei Zimov lighting a Siberian lake on fire, showing how there's a lot of methane in it. Don't worry, he's making it in carbon dioxide, which is lower. Anyway, elephants are essentially the only large animal that's capable of, loves to knock down trees, and hence restore some of the grasslands that were so productive photosynthetically and capable of keeping the ground cold. So we're doing that with a colossal team, and we thank the Zimov team, colossal team, and the Revive and Restore, and a number of other co-authors from our lab. And we also, the Personal Genome Project is the source of almost all of our human data. It's freely available to all of you. It's arguably the only open access trait and environmental and genomic data and sources of cells for the experiments that I described. So my thank you to all those volunteers in multiple countries now. And I finished a little bit early by the clock, but I'll open it up for questions. Thank you very much.
Well, thank you, Professor Church. So we do have a few minutes now for Q&A from the audience, and so we'll just go ahead and run that through the virtual system. So does anybody have a question they'd like to lead off with?
Thank you. Hi, George. Thank you so much for this fantastic presentation. I always get impressed with the amount of science and innovation that can come from your lab. And obviously, all these scientific discoveries that get into novel potential treatments. And when we think about convergence innovation, that's one concept, but I'm wondering how do we think about convergence translation? What needs to happen to get this incredible amount of science and make sure that we can translate it fast to patients? And I'm bringing this thought thinking that we needed to get COVID to get everyone focused in trying to solve one problem. And when we did that, we were able to get to a vaccine. So now that COVID is gone, and on the concept of genomic medicines, again, we have many tools. We've been talking about these many modalities, delivery systems, things that we can do with science. But what is necessary, from your point of view, to really get all this conversion innovation, all this innovation, and make it convergent to translate into actual treatments to patients?
Yeah, it's a great question. Arguably one of the main themes today. So our lab is well positioned both by getting students from Harvard and MIT and having postdocs that want to be in the hub, so to speak, of all this biotech startups and investors. So that's the first start, is to make a safe place for innovation at the student and postdoc level. And then I would argue that they tend to be maybe more highly motivated than average. I mean, we're all motivated, but to translate things. And our lab and a few others, but not all academic biology labs are, but our lab is oriented specifically towards translation. I mean, we try to do basic science, technology, and societal components all in one. If you can get each project to do all three of those, then that's what we're looking for. But that kind of provides a beacon for that sort of individual to come with that in mind from the very beginning, and so they're really thinking about translation from, let's say, from their first day of graduate school. So, and I think that pays off years later when they actually enter the biotech field, typically in the Boston area, although we've helped start companies all over the world. And then the translation depends on scaling up something that is suitable for clinical trials, and that isn't always something that's incremental. The FDA is quite embracing of new things. They were delighted about our knocking out all the endogenous retroviruses. They being bench scientists themselves, they're very responsive to things that are disruptive in that they're never been tried before. So I think if we have a culture of failure is at the academic level, it leads to a culture of great success in the clinical trials. And by failure, I mean like our libraries. We make 1 million members of a molecular library, and if three or four survive out of that, that's a success. We don't worry about the 1 million failures, and that's kind of the metaphor for our lab.
Do we have other questions? Wave your hand if you do. We got one over here, Sadek. I got to say, I got these lights in my face and I can't see people real well.
Yeah, so George, really fascinating talk. 2 questions. I mean, I think this concept of the aging database and then using combinatorial AAV gene therapy to address aging is a really fascinating concept. As you know, aging is a universal risk factor for death. So I guess the question becomes, at what point do you intervene with this combinatorial AAV gene therapy approach for aging? And then second question related to that is why AAV for that particular context as a delivery vehicle? What's the rationale there?
Yeah, I mean, we're aiming for interventions that are as late as possible for all sorts of practical reasons. Bigger market and faster observation of reversal of age-related diseases and their and if we get if we get multiple diseases of aging simultaneously that have little in common other than aging, we're probably getting close to something that's at the core of those 10 mechanisms, the wheel of hallmarks. So as early as possible. The second half of your question was?
Why AAV was the question. Why AAV?
Oh, why AAV? Yeah. So we also published a paper on cytomegalovirus, a mouse cytomegalovirus, and we're exploring other things. I personally like cell therapies as well, because I mean I know they're not, it's apples and oranges, cell and viruses and non-viral, but I think all of them should be explored. The viruses have the advantage that it's very easy to make these mega libraries, and AAV in particular, easy to make these mega libraries to see how to get to specific tissues or to get more general systemic delivery. So also it was the first one of the first approved by the FDA, and it's one of the most popular ones, has low immunogenicity. But there's no fundamental reason why we picked AAV other than pragmatically wanted to focus on the age-related diseases, not so much on the vector.
Hey, George, this is JC. Thank you. I would like you to expand a little bit on the convergence of two of the chapters that you talk about. On the one hand, you talk about massive libraries for actually screening either, in the case of transcription factors, capsids, etc. And then in another chapter, you talk about the ML training, completely devoid from. So how these two chapters converge, and what would be the next chapter of the interaction of the data generated by MyCell libraries together with ML training?
Yeah, so they were together sort of from the beginning. I mean, we used much cruder in around 2004, 2009. We were using cruder analytic tools, but the instant that the AI machine learning looked like it was ready, we combined that with the mega libraries. The mega libraries can be used for anything ranging from DNA, RNA, protein, cell surface. We have a cell surfaceome, differentiated cells. Anything, and the key thing is not so much making the libraries, which was greatly helped by synthesis on chips, but it's the design of a selection that allows you to prioritize them. And what comes next? I mean, certainly if you have the machine learning, really benefits from having big data, and so for some things it's very hard, very expensive to get big data. For example, on outcomes of clinical trials. But here's a case where you can really turn the crank quickly and get a million shots on goal per cycle. That might just take a few weeks, and each cycle you get better and better machine learning input and output.
Well, maybe I could indulge my inner child and just ask you a little bit more about Colossus. When I first heard about that, I thought, oh my God, Jurassic Park for real, the woolly mammoth is coming back. But how are they going to do 1 million edits on a genome, and then what's long-term the goal for that company?
Right. Well, so the short-term goal is actually generating technologies. So it's already spinning off technologies. It's only a year old, and we've spun off a company called Form Bio, which includes a lot of the computational tools that you need for synthetic biology of this nature, and more generally. So that's kind of both their short and long-term play. There is some chance they might be able to help with carbon credits or something like that, but it's not the main goal. There are reproductive technologies that may be valuable, and multiplex editing may be valuable in human, non-veterinary, and veterinary uses. A million edits, it's not clear we need a million edits, but I don't. It's also not clear we can't get them. I think we're going to start very pragmatically with the top 100 or so that are fairly clearly indicated to be involved in cold resistance, and then because we want to help restore high carbon, low human population density regions of the Arctic, where there's just 500 meters of carbon that could turn into methane. But that may be dozens of changes. If you look at the fixed alleles, that is, say, alleles that have gone to homozygosity in all the Arctic mammoth species, extinct mammoth species, and are not that, and those alleles are not present in existing endangered elephants, then those are candidates. And they're fairly small, and it's on the order of maybe 7,000 or so that have really gone to fixation in all the mammoths that have been analyzed. And ironically, I think we're getting to the point where we have more mammoth genomes than we have elephant genomes. We have on the order of maybe 100 mammoth genomes. When we started this, there was not even a single elephant genome. So hopefully that satisfies your inner child.
Yes. Well, thank you. And I think our time is up now. So thanks, everybody. This has been wonderful.